and welcome to lecture number 2. Today we're going to talk about DNA replication. So to do this, I'm going to draw it all out. Get yourself some colored pens and some paper and get ready to draw with me. I'm going to go ahead and turn this around toward the paper. Alright, so here I am with my piece of paper, and what I'm going to do first is I'm going to draw a piece of DNA. So here's one half of my DNA double helix, here's the other half. Pretend that looks really good. Here are the nitrogenous bases going across, so this would be the sugar phosphate backbone here. This is the other sugar phosphate backbone. Trying to show you its twirly nature because it's a helix. And then I'm going to exaggerate this portion, sugar phosphate backbone, sugar phosphate backbone. It's going to open up and kind of become a little bit more linear so that we can look inside of it for a bit of detail. I'm going to label the sugar phosphate backbone. So sugar phosphate backbone, that's this. It's on the other side too, so I could add a label pointing over here. And in the center, I'm going to have nitrogenous bases that are bound together with hydrogen bonds. So here's A, which always binds to T. Here's C. What does C always bind to? Correct, it always binds to G. So we can go in any order here as long as our base pairs line up. It should be all good. Put a G here, which means I have to have a C on the other side. Put an A here, which means I have to have a T over here. That's our complementary base pairing. We always know what this nucleotide is going to be as long as we know what its other half is. So complementary base pairing allows me to figure out this strand based on this strand. Now, my DNA double helix is anti-parallel, which means that if this is 5' on this side, it must be 3' on this side. The other side is going to be the opposite. So 5' here, that means that 5' is on this side, and the 3' is on this side. They're going in two different directions. So one's going this way, and one's going that way. That's what parallel means. I think I spelled anti-parallel wrong. That's okay. You can spell it right in your notes. Don't worry about it. No one's going to know. So, DNA setup. We have our double helix structure. We've got our sugar phosphate backbone. We know that each one of these letters represents a nitrogenous base. I'm going to just abbreviate that. We've got our complementary base pairing between our nucleotides. They're bound by hydrogen bonds, so we can put like a little hydrogen bond there. Cool. Now that we have all that, let's look at this DNA and decide how we are going to replicate it. To replicate our DNA, I'm going to draw it again. So I'm going to move this down. Cool. I'm going to draw the linear structure of my DNA again. My base pair's in the middle. A binds with T, C binds with G. That would continue. So I'd have G, C, A, T, whatever the sequence is. Then I need to pull these two strands apart. So I'm going to pull them apart and they're going to start to fork in separate directions. I still have nucleotides sticking off on both sides. Right? But now my two strands are separate. There's a special enzyme that causes your two strands to separate. I'm going to draw it as a wedge because it's wedging itself in between the two strands of DNA. And this enzyme is called helicase. We know it's an enzyme because it ends in ACE. And I remember that helicase unwinds my DNA strand because it kind of sounds like helix, right? So my helicase is going to unwind my helix. So that's the first step. Now I have my exposed single strands of DNA. These single strands of DNA are really easy to get tangled up, like they're inside the nucleoplasm, inside your nucleus. It's dangerous in there. There are ions, there are enzymes. So we want to make sure that we hold this strand steady so it doesn't get tangled. So we're going to put some other proteins on it. They're just going to grab onto it, just hold it into place. And these other little proteins are called single-stranded binding proteins, or SSBs, single-stranded binding proteins. That's their job. They're going to be on both sides. They're going to hold my single strand of DNA down, single-stranded binding protein, so that it doesn't get all twisted. Super exciting. All right. So I'm going to draw a little bit more of our DNA so we have more to work with. I'm just going to put all A's so I make it really easy on myself to make sure that my complementary base pairing is correct, which means this half had to be all T's. Fabulous. So my DNA has been opened up by helicase, it's being held into place, and on the first drawing we said that this was going to be 3' and this is going to be 5'. That's cool. That's going to help me figure out what the leading strand and the lagging strand are. So this DNA needs to have the other half of it made, because remember, we're replicating one strand and turning it into 2 strands. So let's start over here. The first thing that's going to happen is I'm going to have an enzyme bind to my single strand of DNA. It's going to sit right here. And that little enzyme is going to create a primer. The enzyme that creates the primer is called primase. I'll just draw it as a oval. I know it's an enzyme because it ends in ACE. It's called primase because it creates the primer. A primer is a little piece of RNA that matches the DNA. So I gave these the nucleotide A, and now the complementary base pair of RNA would be U's. Because remember, RNA does not use T. It uses uracil instead of thymine. So now that I have this primer that matches my single-stranded DNA, I can have another enzyme attach onto this and start to synthesize a piece of DNA that matches my original strand. This enzyme that synthesizes the DNA is called DNA polymerase. I'm going to call it poly. DNA poly 3. You might be wondering why it's called 3. They were named in order of discovery, so DNA polymerase 1 was found first, and then 2. This is the third one to be discovered. So DNA polymerase 3 can attach to where there's a primer, and then it synthesizes DNA in the direction of 5' to 3'. That's the direction of the DNA it's making. I know it's making it in this direction because over here I have 3', so the DNA that's matching has to start at 5' because my 2 halves of a DNA double helix are anti-parallel. DNA polymerase 3 will pull amino acids out of the nucleoplasm and put them together. So here it'd put a T, it would move down, it'd put another T, it'd move down, it'd put another T, same thing here, and it would keep going toward what we call the replication fork, where it forked open. Helicase will continue unzipping the strand, and DNA polymerase 3 can just keep going and going and going. Because it can continue, we call this the leading strand. the leading strand. It's just continuously going toward the fork. Okay, so DNA polymerase III continues on. Then we have to do something about this primer. This primer is made out of RNA, and we don't need RNA in our DNA strand. So we have to go through and we have to replace this primer with DNA after. There's an enzyme that replaces the primer. It is called DNA poly 1. So DNA poly 1 will come in and we'll replace the primer, replaces the primer with DNA so that we don't have any RNA left in our system. When that happens, it leaves a little gap between the strand of DNA that DNA polymerase III made and the replacement piece of DNA that DNA polymerase I put in when it removed the primer. This little gap right here, I'm going to just highlight it. There you go. Little gap right there. That little gap needs to be filled in. And that little gap is filled in by another enzyme. It fills the gap. This enzyme is called ligase. Ligase. It comes in and sticks a little bond in between this nucleotide and this nucleotide so that your new piece of DNA that you've made is just one giant chunk of DNA. So that's an important job. We don't want missing pieces or gaps in our DNA. So ligase fills the gaps. That's our leading strand. Let's talk about the lagging strand. The lagging strand is in the opposite orientation. So 5' is down here, where number 3' is up here. That means when primase comes and adds a primer, the primer sets up DNA polymerase 3, poly 3, to add nucleotides in the opposite direction. This time it's going away from the fork. The reason that it's going away from the fork is because DNA polymerase 3 is creating a strand that goes from 5' to 3', so that it's anti-parallel to this original half strand. And as it goes from 5' to 3', it can only add new nucleotides on to the 3' end of the strand that it's creating. So it can only add a nucleotide here, and here, and here. It can't go in the opposite direction. On the leading strand, DNA polymerase 3 was going in the same orientation, but because the 3' strand was leading toward the replication fork, you could just keep adding nucleotides as your DNA unzipped further and further and further. So that means as our helicase continues to unwrap the DNA, I'll get another primer up here, and that means I'll have another DNA polymerase 3 going in this direction. I'll end up with these little chunks of DNA called Okazaki fragments. And that indicates to me that this is the lagging strand. The lagging strand is the one that contains Okazaki fragments. I abbreviated that. And it contains Okazaki fragments named after the scientist that discovered them, because DNA polymerase 3 can only add new nucleotides to the 3' end of the strand that it is building. I'd add another primer, make a little strand. As this opens, I'd add another little primer, make a little strand. Then, because primase makes primers of RNA, I still have to remove the primer, and I have to replace it with DNA. So that involves DNA poly 1. So DNA polymerase 1 will come in and will replace the primer with DNA. That will leave a gap between the pieces of DNA and the primer where it used to be, which has to be filled in with ligase. Ligase will come and fill in the gap, and you will eventually end up with 2 strands of DNA that are identical to the parent strand. 1/2 of the strand is from the original parent strand. One half is new, being created by DNA polymerase 3 and all the other fun enzymes that are involved. And they are identical, which is really important. You want your DNA to not be mutated every time you duplicate it. So this seems like a whole huge process, and it is, but I think it's important to remember that this happens in your cells all the time. I'm doing this for my red blood cells, for my stomach lining, for all of my mucosa membranes, for my nails and my skin, and all of the parts in my body that I'm building. Every time I divide a cell, every time I do mitosis, my DNA has to be duplicated. So a pretty incredible, intense process that happens regularly. And that's the end of our DNA replication lecture. So let me know if you have any questions. Feel free to email me your drawings if you want me to see them. And if you want, listen to this lecture more than once and practice drawing it more than once. We know how important drawing is for learning, so I think it's a good use of your time. Have a great rest of your day. Talk to you soon.